Millions of years of evolution have endowed our bodies with a natural ability to fight disease. Our immune systems have a remarkable and ferocious mechanism to identify foreign invaders such as bacteria and viruses and destroy them. For over a century, scientists have wondered whether this natural mechanism could be harnessed to combat disease. Could a magic bullet be made that could selectively identify and destroy the disease agent itself? At the heart of this process is the antibody. 40 years ago, Nobel Prize winner Cesar Milstein and his team at Cambridge Laboratory for Molecular Biology built upon this basic idea to develop a technique to produce bespoke monoclonal antibodies. Antibodies that are produced by identical immune cells from a unique parent cell. Once monoclonal antibodies have bound to their target, they mark them for destruction by the body's natural defense mechanisms. This pioneering research was the inspiration for the development of a remarkable new drug that had the potential to treat a whole range of diseases, from leukemia to arthritis, cancer to multiple sclerosis. This is the story of Campath 1H. In the late 1970s, Professor Herman Waldman, then a lecturer in the Department of Pathology at the University of Cambridge, and now head of the Sir William Dunn School of Pathology in Oxford, was applying for his first Medical Research Council programme grant.
Immunological diseases represent about a third of the most important diseases in the Western world. And we don't have good treatments. We have steroids, we have all sorts of drugs that just penalize the whole of our immune system and damage our organs, our kidneys, to try to stop immune attack. And the fact the body can do it itself told a lot of us that if we could learn how to harness the way the body does it and exploit the way the body does it, then we are going to be much better off than using these nasty drugs. The field has depended on all sorts of quite ingenious approaches to study what goes on. Because our goal was therapeutic, our approach was to say, can we produce immunological tolerance to something that would normally be a foe, and now make it appear as a friend?
The approach Waldman took was to make an anti-lymphocyte antibody using a technique that had recently been developed by Cesar Milstein at the MRC Laboratory of Molecular Biology in Cambridge. By fusing myeloma cells, cancer cells that develop from B lymphocytes, with cells from the spleen, Milstein had managed to make cell lines that indefinitely produced large amounts of a single antibody, known as a monoclonal antibody. Such monoclonal antibodies were ideal for Waldman's experiments and were given the code name Campath, standing for Cambridge Pathology. An immediate medical application became very clear: to improve bone marrow transplant in patients whose blood systems have been destroyed by leukemia and radiotherapy.
The very first patient the 2B antibody was tested in was a patient with leukemia. And the antibody, we manufactured the antibody in our laboratories in Cambridge. We made enough of it to treat the patient, and the patient was treated at Addenbrooke's Hospital. And it was the first time that we saw leukemia cells being cleared from the blood and really disappearing not only from the blood but from the bone marrow of this patient. It's a very exciting time for us. And I remember Herman coming into the lab and saying, Jeff, it's really working. It's almost like a miracle. It's worked so well, better than we thought. And we were very excited about that. And one of the big problems was that the antibody had originally been made in rats. It was a rat protein. And so if people were treated with it for some time, they started to make antibodies of their own, anti-antibodies, if you like.
Fortuitously, another Cambridge scientist, Professor Sir Greg Winter, had just developed a way to humanize antibodies.
We had the idea that maybe we could take a mouse antibody and just transplant those little bits from the mouse antibody that are responsible for binding to the target and put those into a human antibody. And in that way, transfer the binding activity of the mouse antibody into a human antibody. And that's what became known as humanization of antibodies.
Time went by. We went on working with the Addenbrooke's team, particularly with Alistair Compston and his team working on multiple sclerosis, and with Professor Roy Cowan and his team who were doing kidney transplants, and they were using Campath for preventing rejection of kidney transplants. So there are a number of different clinical trials in different places around the world, but particularly here in Cambridge.
During the late 1980s, it was becoming clear to researchers that MS is an autoimmune disease, a disease in which a person's immune system attacks their own tissues.
So multiple sclerosis is the commonest potentially disabling disease of the brain and spinal cord affecting young adults. The illness targets the myelin sheath, which surrounds nerves in the brain and the spinal cord, eventually damaging both. For people who have multiple sclerosis, the illness usually starts as attacks which come and go, and they're well in between, but later those attacks reduce in number and they're replaced by slow progression, so that in the end the affected person faces both physical and mental disabilities. Multiple sclerosis is characterised as an immune disease for two main reasons. First of all, because if we look at affected tissue, we see evidence for chronic inflammation of the brain and the spinal cord. And secondly, animal models of multiple sclerosis have shown that transferring components of the immune system will indeed reproduce the disease. We began to wonder whether we could help patients with multiple sclerosis by preventing the movement of activated lymphocytes from their bloodstream into the brain and spinal cord.
Treatment with Campath 1H looked like one way that this could be achieved, and in 1991, Comston started an 18-year-long collaboration with Waldman and his team by trying this approach for the first time in a woman who had developed MS some years earlier. By 1999, Comston and a clinical trainee, Dr. Alistair Coles, had treated 27 patients, all of whom had already entered the secondary progressive stage of MS.
We paused to analyse our results, and we realised that although we had stopped the inflammation in the brains of our patients, and that had led to them having no further attacks of the disease, unfortunately their disability continued to progress. That is, the problems that they had prior to receiving Campath just gradually got worse.
This observation puzzled the researchers. If MS is an inflammatory autoimmune disease, why was Campath 1H treatment failing to help people in the progressive phase of the disease, even though the treatment seemed to turn off inflammation?
What we realised is that disability in people with progressive multiple sclerosis continues because nerve cells wear out, they degenerate. And Campath hadn't helped them because it isn't inflammation then and there that's causing this degeneration. We speculated that this degeneration was due to damage done to the nerve cells some years before, during the relapsing remitting phase of the disease, when inflammation is the predominant process. So we concluded from this that for Campath to be effective, we really had to treat people much earlier on in the course of the disease, when inflammation was the main problem, to prevent nerve damage, which ultimately might lead to nerve degeneration.
Patients enrolled in the trial had to meet strict entry criteria and were treated with up to 3 annual doses of Campath 1H. The effects of the drug were measured 3 years after the initial treatment.
And here we saw confirmation of our findings. Treating people early on in relapsing remitting MS results in reduction in their attacks and not only stabilisation but actually improvement in their disability. The next stage of testing a drug is a phase 3 trial, and we compared Campath with active therapy, a standard drug, beta interferon, in 2 separate phase 3 trials. The difference between them being that in one trial we looked at people with multiple sclerosis who had never had any other previous drug, and in the other trial we looked at people who had already been on a standard therapy and had had some breakthrough of their disease. And in both trials we saw that Campath reduced attacks of multiple sclerosis more effectively than the standard treatment beta interferon. And in the second of those trials, we also saw that Campath reduced the risk of becoming disabled and indeed improved disability in people who'd already been on a standard therapy and had had an attack of their disease. And it's this result that is unique. There is no other drug in the treatment of multiple sclerosis that has shown efficacy over and above the standard first-line treatments in this treatment-resistant population.
In 2013, Campath, manufactured under the name Lemtrada by the pharmaceutical company Genzyme, was approved by the EU to treat multiple sclerosis. Now in 2014, it has received approval from the National Institute of Health and Care Excellence. This drug, which has taken decades of research from the University of Cambridge, will finally begin transforming the lives of thousands of people living with MS in the UK and Europe.
Well, looking back over the last 20 years during which we have been developing this programme, it is, I think, a reasonably good example of what I like to call clinical science, or what is now called experimental medicine, in which the approach is to treat patients, to see what happens, and to learn from that experience in order better to understand the illness. We did face struggles along the way. It was difficult to sell the hypothesis and to explain the risks of what is a radical treatment of the immune system professionally, to funding agencies and to the pharmaceutical industry who owned the rights to the drug at different times. And perhaps if those very understandable hesitations had not been there, we might have been where we are now, say 5 years ago. But in the end, the results speak for themselves, and it's for physicians and neurologists to decide how best this drug is used in a market where there are already a number of other treatments for multiple sclerosis.
We always wanted to be able to use the body's killing mechanisms, damaging mechanisms, and the antibody to be able to recruit them to kill what we wanted. And we were pleasantly surprised that that was possible. The second thing that was great was being able to actually go to the clinic, because that had always been my intention. And being able to demonstrate that, in fact, we could deal with some of these unmet medical needs, albeit slowly, and you had to for ethical reasons, but gradually we could see that we were going in the right direction, offering patients something that wasn't available before.
A couple of years earlier, I'd been a basic scientist and then suddenly find myself with this molecule that we'd constructed together being put into patients, and it was terrific. And in fact, that was not only it was transformative in the impact it had on the technology, but also transformative for the kind of my later career development, which was to very much focus on trying to look at other approaches to making therapeutics.
I have been working on Campath as a treatment for multiple sclerosis since 1994, and it has been a huge adventure.
Looking back over the work with Campath 1H, it's been very enjoyable and a privilege to work with so many distinguished scientists and clinicians who've enabled the work. But it's been the contact with affected individuals, the patients, who've put their hopes and fears on the line, which has been so very inspirational.
The journey from fundamental research on the immune system to a commercial product has been a long one. It's a testament to the commitment and determination of the researchers that 40 years after the discovery of monoclonal antibodies in Cambridge and 30 years after the early studies on Campath, we are on the brink of a revolutionary new treatment for MS.